Okay, now if you thought that stratification was difficult in complex disease, how can you actually stratify in a rare disease? And so it's a great pleasure to welcome Professor Tim Cox from Cambridge, who has been working in this area and is leading a consortium on stratification in rare disease.
So Gaucher investigative therapy evaluation is the topic that was chosen. Thank you. So Gaucher disease, a so-called single gene disorder, an ultra-rare disorder, not just an orphan disease, a rare disease, but an extremely rare disease. Allegedly a monogenetic condition described by this Frenchman for his MD thesis, allegedly by genome studies in the population, genetic studies in the population, occurring with a frequency of 1 in 60,000 live births. There are approximately 60 million people in the UK. That doesn't make the cut. It's an enzyme deficiency of the lysosome associated with excess glycosphingolipidoses. If you did medicine or biochemistry at university, you may have omitted to get fascinated by them, but that was a mistake on your part. So the patients may present in this way. As my first patient, a Spanish man, and here is the original description of the spleen by Gaucher, and you can see those large macrophages in the spleen, which are found today with modern stains in the spleen and bone marrow. And it is, to some extent, a macrophage disorder. They arise, as you recall, from transformation of blood cells, monocytes, in the marrow particularly, which enter the bloodstream and then find their way to the various viscera, where they become resident histiocytes and take on the behavior of their neighbors in these tissues, so pulmonary alveolar macrophages, the Kupffer of the liver, and so on. And it is principally, in most cases of the disease, that the phenomenon is detectable. So there is fatigue, growth failure due to an inflammatory response, massive enlargement of the viscera, the liver and spleen. There is anemia, bleeding, and a susceptibility to infections as a result of sequestration of the circulating blood cells in the spleen. And in a proportion of patients, as we now know, Gaucher never did, the patients develop a neurological complaint. In fact, in Asia, more than half the patients with this condition, 1.6 billion people in China, there are quite a few patients with the neurological disease of Gaucher and rare complications elsewhere. Many of the patients that we see who are adults with this condition in Britain have bone disease, and again, it's very complicated. They can either be thin and fragile, we all know about that from Jackie magazine, but there is also painful episodes of death of bone and evidence where the bone is broken down, and I'll show you some images. There's also a cancer of the bone, or cancer that appears in the bone, which is effectively multiple myeloma. So there's a lot going on in this condition, and a great deal to worry about. But if you had the condition yourself, you are at risk from some rather desperate complications. And I'll just show you them here. There's a large unmet need with this condition. So forgive me for giving you the description, but I think I need to put it before you in its completeness. This is an MRI scan of someone with an episode of osteonecrosis, and here you can see the proton signal from the edema in the bone marrow. It's exquisitely painful, as it would be in a patient with sickle cell anemia. Here's a lytic, expanded lesion of the end of the femur, like eggshells fracturing every minute of the day. I'll show you a picture of the patient in a wheelchair at the end. Here's a fracture through just turning at the kitchen sink through the femur. It may be thin bone, but the fracture is equally agonizing. Here's a person held together by metalwork. Here is someone who just coughed and had a fracture through an extreme osteoporotic bone in the spine. And about half our adult patients have had hip replacements early. So there's a big burden here. And then really horrible things come up. Here is a man followed for 10 years on therapy who last January had a painful tooth Eventually, he came to be scanned. We knew exactly what it would be. Here's this dreadful lesion here. We scanned him all over, and you can see there are other lesions in the pelvis that were not present 2 years before. He has multiple myeloma, and he's one of 7 or 8 patients of ours with this complication. It was biopsy-proven. Myeloma sucks. It's a common hematological malignancy. Patients with this disease have a frequency of this 40 or 50 times greater than average. We don't know why. And terrible disease in the liver. And here's the neurological complaint. From a young child, this young lady I saw, now age 14, in the ICU last night. This young baby died by the age of 18 months of age, and this woman here works for the NHS at 52. So you have extreme variation from neonatal, infantile, childhood, and later disease. This is a condition affecting the viability of neurons and nuclei in the basal ganglia and elsewhere. Here, at the end of life, or adult life, we're seeing new onset Parkinsonism. The GWAS scans, funded by various bodies that we all know and have their imprint on this earth, very many millions of pounds, failed to detect that mutations in the causal gene for Gaucher disease is the greatest predisposing genetic complaint for Parkinsonism, not in a selected population of people who get it. It's overrepresented in Ashkenazi Jews, but in all populations: Taiwan, China, Korea, France, America. Astonishing. We never realized that. So this is only just coming to light now, and is a problem in the homozygous form for our older patients. It affects all sorts of interesting people. You understand that it's due to a disease of the basal ganglia, and what's very interesting, that it's associated with Lewy body dementia in Gaucher patients, and indeed you can see that in the core of the Lewy bodies that accumulate in the neurons, there is a mutated form of the glucocerebrosidase gene protein. It's highly associated with aggregated protein in the Lewy body. However, the molecular pathogenesis of Gaucher disease, despite a huge amount of information about the mutations that predispose to it, about 400 known, is not really understood. The enzyme is acid hydrolase. When deficient, it fails to break down these complex glycosphingolipids, which are very bioactive indeed. They become overrepresented. There's little product, and we end up with the phenotypes I've just shown you, but we don't know quite how they're brought about. So it won't be the first time that we try to understand diseases by treating them. In the old days as doctors, we often used to make a diagnosis by treating somebody presumptively in the emergent situation, and how they do tells you what they might have had. I'm afraid that's what we do sometimes. There's also a problem with this disease in that there is variance in its expression even in twins who are monozygotic. This is a pair that we studied here in Cambridge, where one sister had a splenectomy for the disease, the old treatment, to keep the blood counts up, and she ended up with extensive bone disease, and the other sister had normal blood counts, and so on, until she was 80. And here, from the Netherlands, are two twins with homozygous for a very severe mutation that predisposes to one of the neuropathic forms of the condition. One twin had florid Gaucher disease with splenomegaly and with myoclonic epilepsy, and the other twin had nothing of the kind. She had type 1 diabetes. Completely discordant in these twins, who are proven to be twins by DNA fingerprinting. So the concept of an endotype, in other words, very different from classifying disease on the basis of symptom complexes, which really tended to ignore, for practical reasons, the underlying mechanisms, which are of no comfort to the patient, to an endotype which is defined by particular pathways or molecular mechanisms that can be intervened or interdicted by particular treatments. We seem to think that we have distinct endotypes in Gaucher disease. I've shown you some extremes. There are classifications based on the 19th century and the early 20th century. We do believe that there are divergent mechanisms and individual therapeutic responses. It's very new, unlike rheumatoid arthritis, in the therapeutic field, and I was very impressed by how much system you could bring to your study, and I was really amazed by how much has gone on since I was a house physician in rheumatoid arthritis. But in this very rare disease, an orphan disease, the pharmacopoeia was very empty for a long time. We tended to cut out the spleen, as I did in my Spanish man, save his life. He had no platelets. But that is a risk factor. If you remove the spleen, we do know, and about 40% of British patients have had to have the spleen removed by some hapless pediatric surgeon. And the fact is that when that happens, in later life, they go on to develop very severe bone disease, particularly the episodes of painful osteonecrosis. And we showed this in a nationwide study of 100 patients in the UK. 44% of whom had, 44 of whom had their spleen removed in the past. We no longer remove the spleen. So to stratify by treatment, many diseases are heterogeneous, but we hope that discrete endotypes may identify mechanistic targets for interdiction, a nice word, that extensive categorization of genomic and phenomic metadata is underway in principally the acquired or chronic disorders, whatever that means. But these analytical techniques may improve identification of disease processes in a group, but they can't work out the mechanistic phenomena in the individual patient in whom the extreme variation and different activities may coexist, as I showed you, in different compartments. And I think this is really the nature of the approach that will be valuable in the study of this disease, which is an economically important disease, actually, as I'll show you in a moment. So there are evolving therapies over the last 2 decades in Gaucher disease, and I'm fortunate to have been a person to introduce a couple of them in the UK, and that's been very rewarding to a certain extent. So you could restore the population of macrophages that are disturbed, that build up the sphingolipids because they can't digest them as a result of an enzyme deficiency, after having not only had to cope with their own turnover of glycosphingolipids, but those that they acquire by eating effete cells and destroying the blood cells in the normal turnover of blood cells, for example. Or you could decrease the supply of the toxic molecules that I've mentioned to you. You can enhance their breakdown by giving them a targeted lysosomal therapy. That's now possible. You can enhance the activity of the immunogen enzyme, which is very difficult to do with a pharmacological chaperone. You can fantasize about gene transfer at the moment, but you could inhibit, as with the statin, the overproduction of those undegraded sphingolipid molecules to balance degradation and synthesis, which is probably a wise thing to do. And I'll show you what's been happening here. Marrow transplantation for horrible inherited diseases has been going on for a long time. It's unregulated. You can do it. You don't have to go approved through NICE, and you can just carry on doing it, it seems, at the present time, which is pretty amazing, since it costs about £40,000 to do and could be fatal in about 10% of cases. However, it does replace macrophages. And in Gaucher disease, when done, either from umbilical cord blood or now blood not harvested from the marrow, but harvested as stem cells from the peripheral blood after the use of cytokines and growth factors, does lead to, when successful, a peripheral cure of Gaucher disease. A young man here, now 42, coming up, is driving a tractor in the Lake District at the moment. He was in a wheelchair by the age of 4. A pioneering, now deceased, hematologist gave him the first boy ever to be treated this way, a bone marrow transplant from an uncle, and we can find no evidence of Gaucher disease in his peripheral blood, as you might expect, but since that time, his peripheral disease has been completely arrested. No liver enlargement, there was no spleen to get pathology in because it had been whipped out when he was just 18 months of age. So an astonishing phenomenon, but a risky one, and not one that's been done very frequently in this country, still occurs in resource-poor, pompous term, countries all over the world where you don't have to give this expensive therapy. It's been approved now for around 20 years. This extracted from human placentae, 1/3 of the world's placentae, this a recombinant preparation, modified with specific glycan residues, mannose residues exposed to facilitate uptake by the mannose receptor on the macrophages, and which has dramatic effects therapeutically. This is a study where we showed that this agent does in fact go, as it were, after injection in 3 minutes half-life to the affected viscera, outlined here in scanning after injection, just a few minutes after being injected here. And here's the large spleen that had to be removed from this man. There you can see it in situ. And it will shrink the viscera. There's an improvement in well-being, in blood counts. Here's the first patient to be treated in the UK who's now still alive. He was in a wheelchair here, and I was doing tissue typing to see if any of his 3 sisters could be a donor for him at that point. Here we got him treatment, and look, within 3 weeks, his blood counts were normalized. His terrible feet, bone structures were normalized in the feet, in the ankles, and he is now very well. This is a worldwide treatment for 6,000 people worldwide. And, of course, it's huge success. It's hugely expensive. We're hearing about large costs, but these agents are £200,000 per patient per year. There are about 180 patients on this enzyme treatment in the United Kingdom at the present time. So here are the drugs, these are the enzyme preparations. Here's a competing enzyme preparation now owned by Shire, and here's one from a produced in plants not approved in Europe, but which you've used, which is made in plant cells. And here are the companies, all competing each other in this white-hot crucible of ultra-orphan therapy. So Genzyme is there. Genzyme got taken over by Sanofi. Protanix in Israel with the plant cells got taken over for this purpose by Pfizer, and Shire and Actelion are in there. Extraordinary. All competing for that market. Now, this, because it's so fascinating, and because, indeed, enzyme therapy does not address either the neurological conditions, for obvious reasons, and does not repair bone, and does not appear to prevent the onset of B-cell proliferation and, as I showed you, myeloma, there's an interesting way of looking with a small molecule therapy. We were the first to develop this along with colleagues in Oxford in the past, but with a drug that, though approved, was fairly weak and not very well tolerated. But here the idea is to prevent the accumulation of glucosylceramide and the related lysolipid, which is water-soluble and very potent, glucosylsphingosine, otherwise known as cycosine, by preventing its synthesis or attenuating its synthesis, as indeed a statin would attenuate the synthesis of cholesterol by inhibiting HMG-CoA reductase, the first limiting step. So a drug has been developed that is a very highly selective inhibitor of that step, and it's a ceramide analog, and it's an inhibitor in the nanomolar range. It was approved by the FDA as Serdelga in August last year and has received a positive opinion, whatever that means, in Canary Wharf quite recently. We're hoping that all go well, that this will be approved in Europe. Whether or not it'll ever be paid for by NICE and its deliberations, we have to wait and see, but we have patients on the trial. It is a very effective drug. The spleen and liver volumes go down in size, the hemoglobin rises, the platelets go up. And the biomarkers, this one we developed, that one was developed in the Netherlands, which are macrophage proteins that are released, indicators of the alternative activation of the macrophage in this condition, actually come down very spectacularly over the years of administration. What is the gold standard for therapy? What will be the gold standard for therapy? Because this therapy, even though it's a very rare condition and very, very few patients in the UK receive treatment pro rata, however, being the highest leaf of the tree, the healthcare analysts and those who do health economics like to look at it and related drugs. It's very important to work out what we're going to do. And so there were originally 4 Gaucher centres started with a national programme by the Department of Health in 1997. Ours remains the biggest for Gaucher disease in Cambridge, but there are now 8 centres which deal with patients over the UK, referring all over from the Britannic Islands, and about 300 patients are known and seen. And we, of course, look after the patients, the complex multidisciplinary care and all the rest of it, but we also do therapeutic research, and we are now all embodied within a strategic medicine, stratified medicine program, together with my colleague Fran Platt, who is very much involved in the development of the first substrate inhibitor from the University of Oxford. So our milestones are to establish a study cohort. In some ways, this would be very easy. We know each other. We've worked together as a clinical service for a long time. We have done a national projects of research. But you can see the variation that we cope with, and getting a proper database and proper enrollment of our patients requires a huge effort, which we've, I have to say, I'm pleased to say, we're pretty well there for our first milestone in collecting the patients together. The other milestones for our work are discover biomarkers, and we're looking at that. We have some active biomarkers for the peripheral diseases I showed you, which we validated in patients internationally, but we don't have biomarkers and predictive markers for Parkinson's disease and the inflammation in the brain, or indeed to predict who is going to need the appropriate dosing and the appropriate systemic drugs that may have a better effect on the marrow disease. And we need to validate those and to use our long-term longitudinal study to try and understand the matters. This is the Gantt chart, which is always required by any funding body today, and it's a nightmare to produce. And here are the milestones that you can see on it, and here are stratified milestones that are very, very important for us to do to secure the ethics, to understand who we need to work with us, to have a database that's designed to agree the fields which we have, to have the milestones for biobanking, and so on. A critical thing, which is perhaps not mentioned before by Anne, well, it would have been mentioned, but is important here, is that the Medical Research Council, in funding this, is very keen that companies should be brought on board. And we were able to secure an agreement of MTAs by the 3 competing companies. If you think, you know, pharmaceutical industry is very tough, and I know some of you are here, we're very grateful you're here, but if you think that the pharmaceutical world is a tough and competitive one when you're dealing with hypertension, rheumatoid arthritis, I'm sure it is. But allow me to tell you, in the microscopic crucible of an ultra-orphan disease like this, it's hell on earth down there. It's white hot. And I'm absolutely delighted that in the UK, the 3 companies with approval from head office in each case signed up to an MTA to work with us, to give us their expertise in registries, to give us their expertise in data analysis and to help us develop our database, on the first point, and to kickstart some of our computerized services for quantifying radiology and other investigations. So I'm very, very proud that we secured this. It sounds easy, it ain't easy. And we have chosen two main strands of Gaucher disease to study. The bone strand, the osseous strand, which is very complex and causes a great deal of disabilities, I can show you. In some cases, the score for quality of life is a minus number, which is worse than death. I'm looking forward to that. And the neurological score, which of course is extremely disturbing too. So let's take the osteoporosis. We look at osteoporosis, we have DEXA scans all the time. We do know that osteoporosis, thinning a bone with fracture risk, fragility fracture risk, is related to peak bone mass. We do know that there is huge variation in the skeleton of patients with Gaucher disease, and we do know that, in fact, bone mineralization density improves with various agents only when enzyme therapy is given. It's conditional, of course, on correction of the macrophage phenotype. Here are the patients studied over 20 years. We have retrospective data, which is very powerful, actually, certainly in one center, which is our own at the moment, to a very complete level. So we have retrospective data, and here's the healthy range for adults. And you can see that many patients have osteopenia and osteoporosis. We treat them. It's very extreme in some cases, as I'll show you. But actually, a significant number of individuals have bone mineralization density that's way above normal, way above average, even by a Z-score. And here is such a patient, seen here with the DEXA scans here, and here is that bone mineralization density steadily going up over the ages of study. And this tells you that the DEXA scan might be an incredibly useful tool to investigate what's going on in the microenvironment, the microarchitecture, if you will, of the trabecular structure of the bone as shown here. Because what's happening is that bone infarction, bone death, and hypermineralization is occurring within little regions of the vertebral body itself. And when we do the generic score for the vertebrae, we see it rising. It's turning into marble over time. And we're able to look at that, examine it in a highly quantified way with these voxels here, pixels here, and to analyze the trabecular bone score, the integrity of the bone, with a very relatively simple, the analysis is difficult, but a relatively simple technique that we've collected all this time. So we can find predictive master things here enacted within the skeleton of our patients that will be predictive, we hope. So we hope that DEXA scanning and vertebral trabecular bone score will discriminate normal osteoporotic and osteosclerotic vertebrae and predict the occurrence of fracture or vascular necrosis at other sites. And of course, we record that in our database. We have a secondary hypothesis that allowing for the baseline structure, the duration and nature of therapy will affect its evolution. So this is, I don't want to go on, there's a lot of special software which has been collected and put in place with help from one of the companies, not Genzyme, Shire, and we are involved in analysing that. For the neurological strand, we have two projects. One I don't have enough time to tell you about now, but is a genetic project transferred from mice strains which show susceptibility to the various phenotypes of neurological disease in Gaucher disease, with seizures or paralysis occurring, different by their survival, differing by a factor of 10. They either die at 20 days or 200 days, according to strain. And when you map the strain determinants, there are 4 genes which are highly determinant of that. And one of those genes has a missense mutation, and one of those, and the protein is in fact a neurotransmitter. And there is a drug which intercedes that. So we are going to our human cohort to see if we can, those patients that have epilepsy and severe progressive neurological disease on the background of their Gaucher, in fact, can be intervened, and if they have a susceptibility mutation variation in those 5 genes, particularly the one I mentioned. It's a neuroreceptor. I won't go into the details. It's familiar to the pharmaceutical industry. But we're also looking to define the complete spectrum of the neuronopathic disease in the UK. Can we make a true distinction, what's referred to as type 1 or type 3 by sort of 19th century classification? And can we determine the difference between someone who works as a geneticist within the NHS at the age of 52, who's born with a lateral gaze palsy, as you saw, and the little girl that I saw last night at 7 o'clock on the ICU, age 14 years, in a state of paralysis. So can we determine, both of those share the same Gaucher genotype, can we determine what is the difference and how do we pull it out? And can we predict who is going to get the Parkinsonism and the dementia? Which will involve quality of life issues, which are terribly important, but of course it opens up other matters. One new aspect is the discovery aspect for new biomarkers, which I hinted at before I finish, which was that it's possible to study peripheral blood markers that reflect ongoing neuroinflammatory changes in the brain and spinal cord. And we have the idea that variation in genes implicated in strain-susceptible mice, as I mentioned, are predictive. But we also have the idea that peripheral markers will accompany the neuroinflammatory changes in the cerebrospinal fluid and in the peripheral blood with the inflammasome. And in experimental models of other disorders, modeled in mice with single genetic processes, indicate that determinants of the inflammasome and of other determinants of neuroinflammatory responses alter and are predictive for the phenotype. Here is peripheral blood. Here's interleukin-1 beta expression in a neurological condition, not too different. A lysosomal complaint, hydrolase deficiency in the brain, and here's caspase-1, which you can see can be invoked from a peripheral blood sample. So I'm going to finish now. Endotyping, deep phenotyping is needed to define the range of endotypes. A bit of a buzzword, deep phenotyping, but it means very careful annotation of selected determinants of quality of life and likely determinants of pathways of pathogenesis, which will require clinical data describing the continuum between disease and health. There are patients who are homozygous for the Gaucher mutations who never know they have it, only found to have the condition when a geneticist comes along in the 100,000 Genome Project. It also determines longitudinal data to increase the temporal resolution of your studies and in understanding age-specific changes, which Anne was mentioning about in relation to therapy. So we believe that enhanced resolution from initiation through to propagation of the full-blown disease in this project will better enable us to define what is going on in terms of susceptibility, environmental influences, and, as I mentioned, inflammatory responses. The treatment is very expensive. This woman, and it is in public documentation, so I'm enabled to tell her, is a former school teacher from Aberdeen in Scotland. We see her quite regularly. She's been elected 3 times. She's going to be busy this spring, I understand. She is the chair of the Work and Pensions Committee at the present time, so you better all sit up. She was elected in a wheelchair. She remains in a wheelchair. Her spleen was removed when she was far too young. She's very grateful for the treatment that she got, but I'm quite sure that we won't be seeing this level of disease in someone like this ever again, with the help of stratifying medicine and giving the right medicine to the right person at the right time. Thank you very much.
Thanks very much. Time for a quick question. You didn't mention about power and
Yes.
given and stratification.
Yes.
You heard some of the issues in the
I did.
pharmacogenomics
I
and
did.
in rheumatoid arthritis. What kind of things have you done to try to overcome the
Thank
power
you.
issues?
Thank you.
And what kind of statistical techniques are you
Exactly. I
use?
always had trouble with power, yes. And as you recall, sir, that it did come out when we were interviewed in front of the Medical Research Council, and a statistician, Brian Tom, of the Biostatistics Unit, has done a very careful analysis of a power calculation. We do have sufficient power with the therapies that are currently available to determine the stratified treatment responses to the bone disease. I can reassure you that this has been looked at. He gave a presentation. Unfortunately, his handouts were not received. It comes up all the time. He is very much involved with this project. I think he may be involved with your own project, actually, in rheumatoid arthritis. But he is, in fact, he's a shared biostatistician. He is in this ultra-rare area. We feel that of the 250 predicted recruits to the study, it's very popular with the Disease Association at the present time, we will have sufficient power of around 180 to 200 people to make discerning decisions about therapy outcomes and categorization of at least 2 or possibly 3 groups.
Do you want to use more Bayesian methodologies?
Yes, we're quite correct. And of course, people always talk about adaptive designs and so on. But we're quite correct. There's a prior probability issue, number 1. Number 2, it's going to be time to event. So we have retrospect and time to event documented in defined ways, which is actually what we've done with a prior study of 100 patients in the UK. So absolutely correct. It's the critical thing. Can we get a number, a quantification in this very, very rare application? Really. Yeah.
Fantastic. Well, thank you very much. Sorry, Gordon.
I just have, if I may, a very specific clinical question.
Please.
I hadn't realized that myeloma was a late, or could be a late feature. Yes. And I wondered, is there anything to distinguish that myeloma from the common or garden myeloma that people get in the absence of Gaucher's disease?
Common or garden, yes. No, except that it drives fast. So I think about 5% of us beyond the age of 65 have a monoclonal gammopathy, which is the biomarker for proliferation and autonomy of a plasma cell clone in myeloma. And in Gaucher patients, various series, including our own, we see perhaps 30 or 40% by the age of 50. So there's a drive. It's being driven harder, earlier. And as I say, that man that I showed you with the jaw, he did not have an M-band when first seen 10 years ago. He started on treatment. The M-band enzyme treatment did not go away. But he presented then with something I thought, oh my gosh, I hope it's not a dental abscess, it's a plasmacytoma. Unfortunately it was, and myeloma was established. So it wrote very, very fast. And the others, it appears to be that we have young patients. A 38-year-old Polish man presented with myeloma. A young girl died of a B-cell lymphoma in her mid-30s. So the drive is very strong. And we find that in a mouse model, actually, which we have, which is prevented by eligostat, which is just out in press at the moment. It's a very thrilling thing. So I actually think we'll be able to prevent that. It's the sphingolipids. How they do it is very fascinating.
Thank you.
Have you examined the pathophysiology of the bone disease and compared it with the pathophysiology of myeloma? So both macrophages are behaving abnormally in the bone marrow environment.
Not specifically speaking. These patients do have an increase in interleukin-6, which is a strong, it's a macrophage-released, as you know, cytokine, and is a very strong driver for the biology of the B-cell. And, of course, there's an autocrine loop which keeps myeloma going once it's established and proliferating. So in relation to the bone, that microenvironment, and we do know that interleukin-6 in particular, there'll be other cytokines, is very prominently expressed and may be one of the final effectors of the alternatively activated macrophage. In relation to the other aspects of the bone disease, I think they're very different. One is the microcirculation, and the other will be the inflammatory response, which prevents osteoclastic activity and stimulates osteoclastic activity. It's fascinating, but complex.
Okay, well,
Thank
thanks
you.
very much.
Thank you very
indeed.
much indeed.